he was a student in the RU program this past summer, and he's going to be talking to us about the foraging in Eastern Copperheads. We got to give Jordan permission to share his screen. Give me 1 second. I'm not seeing an option to give him permission.
Let me try, Ted. I just hit. Jordan, can you share your screen now, Jordan?
Let me see. Yeah, I can. Okay.
Okay. Thank you, Jordan.
Is everything good on mine? Okay.
Yes, we can see it, Jordan.
Sweet. Okay, well, I'm Jordan Stanton, and today I'm going to be talking to you all about variation in the seasonal emergence of cicadas and its effects on the Eastern Copperhead. I conducted this study under the supervision and mentorship of Reni Gull, Dr. Stephen Richter, and Jesse Sockman. Also completed it alongside assistants to the study, Marnie Behan, who's responsible for the majority of the pictures used throughout this presentation, and Jackson Skodel. So to provide some insight onto why this study was and is important, I'd like to talk about interspecies relations. Humans, as we all know, have a massive and constantly growing population. And rightly so, we commonly come into contact with other species. Some of these species are potentially dangerous to us, and we can be just as potentially dangerous, if not more, ourselves. To minimize the risk of either party being harmed, it's imperative that we have an understanding of both our niche and role within the world and in our environment, and that of whatever species we're coming into contact with. This leads me to Copperheads at Coomer Ridge Campground. To make sure that campers and Copperheads maintain safety alike, it's important to understand why those Copperheads have came to the campsite to forage. So I'd like to introduce the subject of our study, the Eastern Copperhead, or Echistrodon contortrix. It's a common pit viper, endemic to Eastern North America. It's nocturnal during the summer, so it forages at night and rests during the day. Any other time of the year, it'd be vice versa. It'd be diurnal, forage during the day and rest at night. It relies on cues to track and capture its prey. Typically, when going about capturing its prey, it relies on passive foraging techniques or an ambush coil, to be specific. So a Copperhead would sit and wait for its prey to pass it by so it could strike and envenomate it, and then later track the weakened prey and eat it without any struggle whatsoever. But they're also capable of utilizing active foraging techniques, where they simply search and capture their prey without sitting in wait for them. Despite what type of foraging technique a Copperhead utilizes, whether it be passive or active, it will always rely on at least one or more of its sensory systems to track down that prey, whether it be sight. By that I mean the ability to pick up on light differentiation, hearing, the ability to pick up on vibrations on the ground or in the air, chemosensing, picking up on chemicals on the ground or in the air, thermal sensing through the use of the thermal pit, which allows them to pick up on organisms that have a significantly different internal temperature in juxtaposition to their environment, and taste, which is sort of a last resort. Like if a Copperhead has tracked something down and wants to know more about it, it'll eat it just to see if it provides any benefit for it. And on to eating, preferences, their diet. In Copperhead's diet, it's large and diverse, and it's mainly dependent on location and ecosystem, as well as the season. So Copperheads while this is not a phenomenon by any means, this happens all the time. Copperheads at Coomer Ridge during the summertime prefer to eat cicada over anything else. And it's unknown as to why. Further studies are being done on that, but they certainly have an affinity towards them. But to add more context of the place I've been talking about so much, I'd like to introduce Coomer Ridge Campground. It's located in Daniel Boone National Forest. It's host to multiple campsites and hiking trails, and it has a wide array of plants and animals in it. Even so, the majority of our focus was contained within inside the primitive site, which is highlighted in blue. It's camp or walk-in sites 46 through 50, and the adjacent bathroom that rests in the middle of the campsite. To give better context of what it looks like, here's just a picture that doesn't depict the entire site, but a part of it. So this main trail would lead into it, and these small, thin trails all lead, or except this one right here, lead to other trails or the restroom. To give further context of the system, I'd like to go through its history. It was established by state and federal agency personnel, and was adopted by Eastern Kentucky University in 2016, focused primarily on local copperheads' relationships with cicadas, and it had a seasonal research period only during the summer because that's when the cicada emerged, and it was also the best time for copperheads to come out and forage due to the temperature being higher. Cicada data wasn't taken until 2018, so there's no data for 2016 and 17, but from 2018 onward, it has been taken. And that data shows that there's a variance in number of cicadas and copperhead each year, and it brings up the possibility of a correlation between the two. So with that information in mind, I set out some objectives for myself with the help of my mentors. And they were to determine if there was a relationship between cicada and copperhead abundance, determine if the relationship fluctuated throughout the years, and if that abundance was influenced by environmental factors such as temperature or humidity. And lastly, to determine if the heat signature between cicadas and their environments was significant enough to allow the copperheads to locate them using thermal cues. So to fulfill these objectives, I had to go through a set list of methods, some of which were already a part of the system already, like taking the environmental data. Before searching for any specimen, we would take the temperature of the ground, the air, the humidity, and we would also take moon illumination. And after that, we'd transect the site for activity, whether it be copperheads or cicadas. And when copperheads were found, they were scanned for pit tags. And if they didn't have pit tags, they were taken back. If they had pit tags, they were dealt with according to if they had been found that night. As for cicadas, when they were found, the data that was noted was their position, if they had emerged from their exuvia or shell, and if they had been consumed by a copperhead or not. At the end of the season, the nightly counts for cicadas and copperheads was compiled, as well as the average nightly temperature, moon illumination, and humidity. The total counts were run through Shapiro-Wilks and QQ norm tests to determine if the data's distribution was normal. A Kruskal-Wallis to show if the counts were dependent on the year, and a post-analysis to determine if there were any outliers. After that, the results are ready and available for us. So here they are. For the nightly cicada counts, it's clear to see that 2019 had the highest count of any year, with 2018 being the second highest and 2020 being the lowest. 2019's didn't start as early as 2018's, but it hit a higher peak and maintained higher numbers throughout the time that it ran. 2020 had the lowest numbers and started the latest in the season out of any of the three. As for annual nightly count averages, as you can see, 2019 was the highest for copperheads. Second was 2018, which is similar to both 2019 and 2020, with 2020 being the lowest. As for annual cicada averages, 2019 was the highest and significantly different from 2018 and 2020, both of which were relatively low. But from the averages, it's clear to see that there is some sort of relation between the number of cicadas present and copperheads present. In terms of the temperatures relation to the two, there's no significant difference between any of these temperatures, but something of note is that 2019, the year with the highest count for copperheads and cicadas, was also the lowest year for temperature, sitting at 22.4. And it's actually in inverse to the two nightly count averages. The highest year wound up being 2018 with a average temperature of 25.2 degrees Celsius, and in the middle was 2020 at 25 degrees Celsius. As for average internal temperatures of cicadas, a lot of the data on it was inconclusive and couldn't have been rounded up into very decisive numbers for the fact that the counts of cicadas were very low. Only 16 cicada were able to have their internal temperature taken. And they're all listed here. They're also listed with their sex, denoted with an F or an M respectively. Internal temperature is listed in blue. Air temperature is listed in orange. Temperature of the tree they were found on, all of these were found on trees, just to let y'all know, is denoted in gray, and ground temp in yellow. That's a little hard to decipher. The internal temperatures of females was generally higher than males, and it was generally higher than the temperature of their surroundings. Males, however, rarely had temperatures higher than females or of even their own surroundings. And I'll go into why that is in 1 second. But like I said, going through these results, it was easy to see that there were similarities between the cicada and copperhead averages. Once again, in terms of cicada count averages, 2019 was significantly different from 2018 to 2020. And in terms of copperhead averages, 2018 was similar to both 2019 and 2020, residing as like a median between the two. Further data is needed on the relationship between copperheads and cicada counts and temperature. They're inverse. Once further data is taken, it'll be much easier to decipher if there's a strong correlation between temperature and the presence of cicadas and copperheads. In terms of future work that could be done with this or within the system, looking into other environmental factors aside from temperature or alongside temperature would be helpful. Precipitation and humidity especially. Cicadas have a much easier time emerging from their exuvia when they're moist, so that might make them more likely to emerge and appear in a higher in abundance. And despite the fact that precipitation would make the temperature lower, a copperhead would still be relatively likely to come out if more cicadas were present. Internal temperature and the sex of cicadas is also another thing that's pretty worthwhile to look into. Like I said earlier, females tended to have a higher internal temperature than males, likely because males have a hollow thorax, while females have a thorax filled with eggs. And this difference in heat between the two could make females more easily noticeable to copperheads due to the fact that they have a higher internal temperature in comparison to their surroundings. Other factors could also come into play there, in terms of chemosensory things, but that also is something else worth looking into. The other cues that copperheads utilize to track cicadas, whether it be sight, chemosensory, picking up on the pheromones that cicadas emit, whether they can hear their calls or not, and so on. But knowing which cues that they rely on would definitely be a great help in determining what makes them so appealing and what makes them more easy to find for copperheads than other prey. And that leads me into another point, or my final point. If copperheads are optimally foraging when they choose to feed on cicadas, it's unknown whether or not cicadas have a higher net caloric value than other prey that copperheads also forage for. But copperheads seem to have an affinity towards cicadas. So it's expected that cicadas might have a higher net caloric value. There could be other factors that come into play, but optimal foraging seems like the most likely. That's why more data is being taken on that by myself, Dr. Stephen Richter, and Dr. Luke Dodd. We've been using bomb calorimetry to bomb different specimen. My main focus, and that of Dr. Richter's, is more towards cicadas to see if they have a higher net caloric value. And after we bomb them and take their caloric value, we're going to differentiate theirs and compare it to that of other prey that copperheads feed on to see if altogether they have a higher net caloric value. Most of the data for this study, however, is very inconclusive, and it's going to be some time before a presentation or any information can be shed on that. In passing, I'm very thankful to have had this opportunity and would like to give my acknowledgements to Dr. Stephen Richter, Henderson Gull, Jesse Sogman, Marnie Behan, Jackson Skodel, Dr. David Brown, Dr. Kelly Watson, the NSF RU program, and EKU for the opportunity they've given me. Any questions?
Jordan, I have one question. Pat Calley here. So it sounds like the copperheads really, if you wish, prefer the cicadas as a food source.
Yes.
I know you haven't had a lot of field data, but is there a sense that is copperhead population density, do you think, maybe tied to cicada density? That is, if the cicadas decrease in number, do you think you might see a decrease in copperhead numbers over years? Or can copperhead switch to something else as far as a prey item goes?
From what I've seen, I'm not saying my answer is absolute fact, but from what I've seen, I think the copperheads could very well switch over from the amount of mice and other rodents present throughout the place. I think they could easily. I think it's just all preference.
Okay, thank you.
No problem.
I had a quick question as well. I was wondering, in terms of optimal foraging, it makes sense definitely to look at the caloric value of cicadas, but there's the other side of optimal foraging is energy expenditure. So have you guys thought of a way to, I guess, characterize the amount of energy spent in a sort of sit and wait foraging scheme? So, you know, passive foraging versus active foraging in the context of cicadas, because they have to climb a tree, presumably, to get to the cicadas. So I think the trade-off there is important. I was wondering if you guys had thought about that.
We have. There hasn't been much testing involved, especially in terms of passive foraging. And with active foraging, we had thought about tracking the copperheads to see how long it took them to locate a cicada and the distance they had to travel to find one. But even then, there are a lot of factors that can come into play that could ruin that. I think it'd definitely be worth something that's worth looking into.
Thank you. I think it's interesting, and I think there's a lot that could be done with that. So that's really cool.
Just to tack on something extra, I was going to ask the same question about foraging energy that Alex asked. You know, the one thing you could use potentially as at least part of the equation for the sit and wait strategy, there's probably some estimates of basal metabolic rates for vipers, if not copperheads specifically. So at least that's probably part of the equation that you wouldn't have to measure yourself. Of course, the whole how much energy are they spending when they're climbing is the other half of that. But I suspect there's data out there that are at least somewhat close for how much energy is expended just in the sit and wait strategy.
I'll definitely make sure to look into that then. It's completely worthwhile in understanding the whole relationship between the two better.
Question, one of the things you noted was that if you encountered a snake, you checked to see if it was pit tagged, and if not, then you did mark it. From just your data there, are you getting, is it a small number of copperheads, the same individuals, or were you getting a lot of different individuals coming into this area as the study went on?
It mostly depended on what night we were going about. Like early in the season, we would encounter decently new individuals, but as time went on, the same ones would show up along with a few new ones every now and again. For the majority though, it was usually those that came in repetition that we found.
Sorry, Jordan. I've got a question for clarification. This is a little bit outside my field, but so did you say that the alternate prey would be like rodents, like mice, are kind of one of the most common types of alternate prey? And then if so, are you saying that your hypothesis is that a cicada would offer more caloric, like would provide more calories of energy than like a rodent would?
Okay. Rodents are just one of the examples. They also eat other insects like the larvae of moths and butterflies, as well as small reptiles and amphibians. But what I'm saying is not one cicada in comparison to one mouse, but overall. So if a snake was able, or if the copperhead was able to capture and eat a succession of cicada with more ease than it would in finding a mouse, that would be a greater net caloric value.
I see. That makes more sense. Thank you.
No problem.
Hey, Jordan, I have kind of a different type of question for you. Seeing you kind of get started with the RU last summer, I feel like you've kind of grown a lot and learned a lot over the last year. And I'm just wondering if you can kind of reflect on, as a developing scientist, kind of what are the challenges and what are you kind of enjoying most about doing science and learning how to do it? And what are you seeing as kind of the challenges that you're facing that make it, hard to become a scientist?
I think in terms of what I enjoyed the most and the biggest challenges are, above anything, I'd say the freedom of science. I guess it's great to have the ability to research what I'd like to, explore the things that interest me. But that freedom also gives into the leeway of being able to look into much and spreading yourself too thin. And I think one of the most important things that I have to do as a person is maintain focus within my study, just to make this as an enjoyable experience as possible. But I definitely am happy to be doing it.
Jordan, the focus is a problem a lot of us have, quite frankly. And if you find science fascinating, it's like, where do you stop? And so what you're saying is you've got a very inquisitive mind and a very curious mind and also a good work ethic. So it's a nice weakness to have, being unfocused. So good for you.
Thank you. I'm glad I'm not the only one.
Then, Jordan, I had just a quick question. Just kind of something that came to me. As far as what do you think might be attracting copperheads to cicadas? Cicadas are some of the loudest insects you're going to find. Do you think, and I don't know much about, reptile, auditory senses, but do you think that their sort of mating calls in the males are sort of maybe attracting copperheads to them? That may be one of the main factors.
I think it could come into play. From what I've seen, cicadas typically only call during the day. As they emerge at night, they have to rest and let the blood flow into their wings and the rest of their body. And during that time, they're very vulnerable and don't do very much of anything. So when they're most available is at night, and that's when they make the least sound. Over anything, I'd guess it was chemosensory, and it can also be thermal. We still don't have enough data either way, but sound could definitely be a factor nonetheless.
Okay, cool. Thanks.
Are there any other questions for Jordan? All right, thank you, Jordan. Great talk. So next we have Reni. Presenting, let's see. And I don't think.
Yeah, Reni, you should be able to share your screen. Can you try it, sir? I've got multiple screen sharing activated.
Okay.
We're good.
Is it working?
Yes, it is, Reni.
Yes.
We see
Great.
you.
Everyone could see my email. Fantastic. All right. Is this visible for everyone?
Yes.
Cool.
That's good.
All right. So this is my undergraduate senior research. When I was an undergrad, I actually did marine biology instead of herpetology, although my mentor at the time, Terry Farrell, also had me working with rattlesnakes too on the side. But I developed a really strong love of crustaceans as an undergrad, and so I wanted to share that with you guys today. So this is my senior research project, which was predation success and handling strategies in the Florida black-tip stone crab, Menippe mercenaria, in foraging trials with 4 different shelled marine invertebrates. And to start off with, I approached this project from predator and prey coevolutionary theory, which is essentially the idea that predators are going to adapt to better hunt down their prey, and prey are at the same time going to adapt to essentially reduce the effectiveness of those predatory adaptations. And it becomes this kind of circular cycle, like you can see in the photo on the right, where it just goes in a circle of adaptation to adaptation to adaptation. And this has been hypothesized to lead to essentially selective pressures and overall nullification of population loss that causes long-term persistence within most ecosystem types, which is pretty cool. And then it's also been postulated as one of the reasons for the increasing complexity of organisms over the duration of life on Earth. And so there are many different types of adaptation and how they affect organisms. I'm going to talk about 2 primarily today, which is behavioral and morphological. So for morphology, a predator's morphological adaptations are going to generally aim at decreasing prey handling time in some way. With the prey item, its morphological adaptations are usually going to be some form of physical defense from predation to increase that prey handling time instead. For behavioral adaptations of predators, they're aiming to efficiently overcome prey defenses, but instead of doing so through brute force, more finding an actual way to overcome it in terms of a clever strategy or behavior like ambush predation, for instance. And for the prey, their behavioral adaptations will generally become focused on avoidance of their predators or causing predator vulnerability. The classic example for this being frogs doing alarm calls when being eaten by snakes. If the snake has its presence known and a hawk sees and decides to swoop down and get it, that frog is going to live a little longer life just because it was screaming a lot. So there are a lot of varieties of adaptations that facilitate this kind of cycle of evolution. And so that brings me to my study organism, Menippe mercenaria, the Florida black-tip stone crab. One heck of a crab. So a really cool thing about these guys is that they're a very commercially important species along the southern coast of the US because they're delicious. Florida black-tip stone crab claws are essentially considered a delicacy. And the really cool thing about them as a renewable resource is that they're renewable on an individual level, not on just a population level. Usually when you're talking about some kind of marine fishery, you're going to be looking at population retention over a period of years while catch is being taken out of it during each seasonal fishing outing. But with these guys, you can actually seasonally harvest the same individual multiple years without killing them. And so the way it happens is a bit barbaric. Essentially, fishermen catch them in crab traps, take them out, and then rip both arms off of them and then throw them back. And these guys, for that, actually have about a 45 to 50% survival rate into the next hunting season, and will grow back their claws during that time. So you can actually have individuals be a replenishable resource. And like I said, that's a really rare thing, and it's really cool with these guys. They live along the coast of Florida, and you can basically find them just hanging out pretty much everywhere. One of the reasons that these guys have such delicious claws is because they're highly specialized as crushing claws built to deal with any kind of shelled prey item they can essentially get their little claws around. And for such a commercially interesting species, we know almost nothing about them in terms of management and life history. Ecologically, a lot of things regarding them are still a complete mystery to us. Almost all research on them focuses just on commercial value and essentially whether or not they die after, you know, their claws are ripped out or they'll survive into the next season. And this is especially the case for juveniles of this species, which are defined commonly in literature as having a carapace width of under 60 millimeters. And so I decided to learn a bit more about these guys for my senior research and contribute to that gap. So looking at these guys, they have dimorphic claws. They are handed, which is pretty cool. And so they have two types. On the left, you have a crushing claw, which is adapted to do exactly what the name implies, crush. You can see right here that there are these two dentae coming out of the main chelae on it, and these are essentially built to take advantage of the idea of minimizing surface area allows you to apply maximum pressure to a point. So once they get something in that claw between those two dentae and start pushing down, it applies basically the most efficient version of pressure possible for that animal structurally. And then on the other side, you've got your fine manipulator claw. This is more of a claw that has small dentae that are serrated. And so they use this for cutting as well as peeling back pieces of shattered shell. With these guys, like I said, they live all over the Florida coast, but you're mostly going to find juveniles within coastal oyster beds as well as mangrove oyster beds. And oyster beds are really cool. If you haven't walked through one while on vacation in Florida, I highly recommend it the next time you're there. They're a very biologically active and nutrient-rich environment, and you will find all sorts of organisms there, ranging from barnacles, sea urchins, starfish, all the way up to sharks and sometimes even dolphins, depending on if the tributary is big enough. So it's really, really great if you're a small predator like a crab to be hanging out in there. And so we did a survey for 4 different types of prey items that we thought they might feed upon as juveniles. The first one is the ivory barnacle, Balanus eburneus. Barnacles are benthic crustaceans. They secure themselves to a surface, have a wonderful calcium-plated shell that has multiple overlapping plates to protect them, as well as a single bottom plate to help secure them to the substrate. And then they have essentially a specialized arm that serves as a covering to the top of the shell to protect them from predators trying to reach in and get them. The next prey item we found was Echinolittorina jamaicensis, your litterine snails. These guys are pretty much your generic small intertidal snail. You'll find them everywhere. They are cute as a button. They are characterized as having a high pointed spire body on their shell with smooth whorls, nothing too particularly defensive about them. They have a very thin shell thickness, especially for such a small size. You held one between 2 fingers, you could probably easily crush it on accident. And of course, like any self-respecting gastropod, they're also going to have an operculum to seal the shell opening. The next we have is Neurita tessellata, the checkered neurite. These guys are also really popular in the aquarium trade, but they are basically characterized by having a very large semicircular shell that is heavily thickened with both interior and exterior ribbing, which you can see quite visibly in the pictures. Their operculum also is secured by additional interior ribbing to make it harder for predators to successfully grasp near the operculum when trying to manipulate them. And with their body growth of the shell, it's also quite unique because they actually grow horizontally instead of vertically, which is fairly uncommon compared to a lot of other marine snails in intertidal environments. So they have a very thick, very defensive, durable shell compared to the other species that we collected. And then lastly, it can't be an oyster study without some kind of bivalve that's included from the environment. So we've got Brachiodontis exestis, the scorched mussel. As a bivalve, it's characterized as having a two-piece shell held together by the abductor muscle near the end of it. These guys have a rigid shell structure, which helps a bit with deterring predators in terms of handling it, but for something big enough, they're not going to have any trouble really manipulating it at all. And these guys also were fairly thin in terms of shell thickness, definitely being thicker than the Littering snail, but thinner than the barnacles and the neurite snails that we also used in the study. So I had 3 hypotheses for this study. First, the trial outcome of whether or not a prey type would be successfully predated upon is dependent on their shell morphology and how that interacts with the structural morphology of Menippe mercenaria's claws. 2, the carapace width of Menippe mercenaria will correlate to the overall prey consumption success, and this goes off of the idea that predators generally will tend to target smaller prey items, and there are lots of classic variations on this theory. For terrestrial animals, gape size is a really classic example. For crabs, it's more often going to actually be claw size, but because these guys had fairly variable claw lengths, we ended up going with carapace width instead. And 3, we also expected between the 1-clawed crabs that we collected and the 2-clawed crabs that we collected, there would be a difference in average trial success numbers between the 2 groups. So for our sample of crabs, we had juveniles that ranged from 15.8 millimeters in carapace width on the right to 42.83 millimeters in carapace width on the left. So we had a pretty good size variance. Each of these guys were housed within a Tupperware container in a fish tank, essentially in Tupperware apartments. They loved these apartments very much and refused to leave them most of the time. 3 of the 18 crabs that we were able to catch were missing 1 claw, so we wanted to account for that within our statistics and experiment. And we collected all of these guys from the Oyster Beds of the Halifax River in Florida. And prior to doing our experimentation, we denied them food for 5 weeks to ensure that we would get positive feeding responses from them during trials. So for the prey trials, each crab got 4 trials, 1 trial of each prey type with 90 minutes per trial so they could acclimate to the presence both for the prey item as well as the crab and then do their thing. Each crab would be randomly exposed to one of the prey items each trial. And so based off random selection, no crab really had a similar degree of arrangement in how they were exposed to the prey items within the trial number. And then we filmed some of these trials as well as collected all of the shell fragmentation from each trial to help indicate the observed handling strategies that they used. For our statistical methods, we used a chi-square test of association for prey type to trial outcome, a 2-tailed Ttest for average prey consumption success between 1-clawed and 2-clawed crabs, and then a linear regression to see if there was a relationship between carapace width and the total number of prey types eaten. So with our consumption outcomes, Littering snails were eaten 15 out of 18 times. Barnacles and mussels both had successes on 11 out of 18 of the trials, and then there were no successful predation attempts with neurites. And so, of course, we found that there was indeed a dependency of prey type influencing the trial outcome. With our Ttest, we did not find a statistically significant difference between 1-clawed and 2-clawed crabs. This might be because that we had such a skewed number of crabs compared to the 1-claws, which was just a byproduct of the random sampling method we did when catching them. That being said, though, this result is actually supported by a behavior that we also recorded and noted during the trials themselves. So that's kind of cool, and I'll talk about that in a bit. And then lastly, in terms of carapace size to the number of successful trials, it was definitely not a linear relationship. We didn't have the time to investigate it at the time, but we strongly suspect if we had looked for a nonlinear relationship, specifically something parabolic, we probably would have actually found essentially that these prey items we used were ideal for a certain size range of, roughly 20 to 35 millimeters in carapace width, whereas those who were below that probably predated on even smaller prey items, and those that were in the 45 to 50 or above range likely switched to a larger group of prey items in the ecosystem instead. So in terms of discussion, crabs ate all of their prey items aside from the neurite snails, which you can see on the right. We believe the reason for this is that neurite shell structure and aperture lip thickness made it incredibly difficult for them to get a strong hold where they could apply successful pressure to the actual shell in the first place. For the littering snails, on the other hand, who were most predated upon, we believe that was likely due to their small size, thin shells. Essentially, they're the molluscan equivalent of popcorn in a lot of ways, crunchy and delicious for any crab. For prey handling techniques, the variations we noted were with barnacles. They would either tear a single side plate off of the barnacle cluster and then just extract your barnacle interior from it, or they would completely deconstruct it very meticulously and leave every single plate intact in a pile on the bottom of their container. For mussels, most of them would just completely shatter it with that main crushing claw. We had, I believe, 3 trials where they also basically decided to turn the mussel around so the hinge was facing them and then cut out the hinge with the fine manipulator claw and then used their mandibles to extract and basically suck the rest of the mussel's visceral mass out through that hinge area. For the Littering snails, like I said, molluscan popcorn, they were basically just crushed completely. And then the neurites, what we consistently noticed was that crabs would be able to pick them up. They would drag them around, very frustrated, and essentially start bashing them along the sides of their container. Being plastic Tupperware containers, that didn't really do anything. However, it's possible if it was a rock or some kind of hard substrate that they were bashing against instead, that might actually have done enough damage to allow a successful predation attempt. So with these guys, as a commercial species, they should really be more well-studied than they are, both in terms of just general knowledge as well as for management. If we are actively harvesting them during catching seasons, then we should understand what management choices are benefiting them as well as not benefiting them when we make those for other species in their same ecosystem. And it's really important to do that to make sure they stay as a sustainable food source, because it's very rare to have a marine food source that is sustainable on an individual level as well as a population level. Another thing we suspected based off surveying and the outcome of these trials is that juvenile M. mercenaria may also be responsible for the distribution of these prey items, primarily in the higher levels of intertidal zones. So you won't find most of these barnacles, especially in the lower areas. Another thing we found was that juveniles would not feed outside of a safe zone, so they always required some form of cover. We originally used an arena for all trials, and they would just sit in the corner and do nothing every time. And so eventually I accidentally came upon the solution of feeding it to them in their Tupperware container where they normally hang out. And from there, everything worked quite well. The other thing we found was that for the one-clawed crabs, they tended to actually use the first two legs on either side of their body as additional manipulator appendages to make up for losing an arm, which was really quite cool. And we believe that's why we didn't detect more of a difference in terms of overall prey trial success between one-clawed and two-clawed crabs. So for acknowledgements, I want to thank my advisor, Terry Farrell, my academic advisor, Dr. Work, our postdoc, Craig Lind, the Farrell Lab, who helped me with trying to get fresh seawater to campus by car. We all know, the phrase life finds a way from Jurassic Park. Well, water finds a way too, specifically to get all over your car. The Stetson University Biology Department for funding, and also Crab A for when it decided to pinch me, to just leave me with a really painful indented black bruise and not actually fracture my thumb at all. That's some of my literature cited. And then if anyone has questions, feel free.
Reni, may I?
Yes, go ahead.
No, I just was wondering, was there in taking a look at like each item that you noted a few differences there, but was there a general consistent behavioral pattern in how they would attack, a particular prey item, or was there a wide range and variety of how they kind of go after these guys?
Usually it was pretty consistent. The different morphology between them did have different approaches, just because, one shape won't fit a claw for them exactly the same way as another. The most variance was with the neurites. Like I said, they basically just started bashing it against nearby objects because they couldn't physically get it all the way under their claws. But probably the two most similar were the mussels and the littering snails, because they were able to successfully just get those straight into the middle of the claw and then clamp down. But there were variations, especially with the barnacles, that they approached it a couple different ways, which were very interesting.
Reni, very entertaining presentation. Thank you. The use of humor is very, very appropriate and very useful, especially in science, I think. This might be outside the scope of what you were doing. So you mentioned the arms race, if you wish, between prey and predator. In terms of the shell thickness of some of these bivalves and whatever, did you notice any big difference in thickness of shells among the animals you were collecting in terms of the prey animals? Or was that just outside was that outside the scope of your study at this point?
There were definitely differences in the thickness between all four of those prey items. And I mean, I know, like, I love to keep a tab on these guys just because I have a secret love of crustaceans in general. And there was, I believe, a journal article that came out last year talking about essentially earlier descendants of these crabs are hypothesized to be one of essentially the big causes for strengthened, thicker shells in a lot of marine snail species today. So there's definitely some interaction there between all of them in facilitating essentially, you get a stronger claw, then I gotta get a stronger shell, change the shape, so maybe it doesn't fit as well. There's a lot of variety that goes into it.
So there's some evidence then, you're saying, for avoidance of being crushed, driving some selection then, or being a selective force.
Oh yes, absolutely. And so, like, with the crabs themselves, they're handed, so you'll have, basically sometimes the crushing claw is on the left, sometimes it's on the right. With snails, you can see the same thing, where there's usually some variance in the direction of torsion in the shell as well. So sometimes you'll have it spiraling to the left or spiraling to the right, and that difference can effectively nullify the use of like a claw to be able to grab onto it completely, depending on the species.
Wow,
which
that's
is really cool.
That's neat. I think Jenny had her hand raised. Jennifer?
Yeah, I just wanted to kind of add to that. So I know that there's limited research, I should say, on this species, but in crabs of similar sizes, are you familiar with any like biophysical research as to like the force associated with their claws? I know we talked about morphological like structure. I know there are a lot of things, physics research out there. Well, you've worked with crocs before, so I think it's like upwards of like 16,000 newtons or something of force with them. Do you know, have similar studies been done on crabs?
I don't know, to be honest. I mean, I know we kind of touched on this like last year with the crayfish seminar, that crustaceans in general are kind of understudied at the moment. I can't say I know any off the top of my head for just straight up like force measurements, especially for these guys. I can give the not so specific analogy of I had one that bent a metal probe I was using to help handle it in like just straight like that. So there was definitely a good amount of force that they could exert. And that's part of why their claws are so delicious. All that muscle mass. It's great.
Nice. Thank you.
Like popcorn.
Any other questions?
Reni, I apologize if I missed this during the talk, but so you mentioned that with the other type of snail, that they were trying to bang it against the side of the tank. Do you think that then you're underestimating the amount that those are actually eaten in the wild based off the terrain in their natural habitat? Do you think the crabs are going out and trying to eat these in the wild and banging them against rocks to help break open the shell, or no?
I'd like to say yes, but I have no basis for it outside of just what I witnessed in this specific study. But I do think it was quite violent. So I had them like basically stacked in little towers within the aquariums to maximize space. And a couple of the ones doing it actually toppled the towers in the process. So it was a good amount of force behind it sometimes when they were doing it. So I think if the surface was like just strong enough and, thick enough, something like a rock, for instance, I don't know if it would have been totally successful, but it definitely would have probably hurt for sure.
Okay, thank you.
Any other questions? All right, thank you, Reni, for that great talk. So unless anyone has any